Right. All along, we have been talking about competition, which is a weed and crop interaction. Now we're going to talk about another interaction known as allelopathy. Now, the aim of this lecture is to understand the role of allelopathy in crop-weed interactions. Hopefully, by the end of this lecture, we should be able to define what is allelopathy. We should also be able to understand plant allelopathic effects, for example, allelopathic effects of weeds on crops, that is A. B, allelopathic effects of crops on weeds. C, allelopathic effects of weeds on weeds. D, allelopathic effects of crops on crops. Then the third objective is to be able to understand how to study, how to investigate allelopathy. So there are certain research methodologies which we want to look at, which can demonstrate allelopathy. Now, if we take a plant, we can divide it into 3 parts for convenience. We've got leaves, stem, and the roots. Now, you may be interested to know that the plants contain chemicals, which are known as the allelochemicals. Most of those allelochemicals, we don't understand their metabolic function. When they are within the plant, they are just produced by the plant, but we cannot identify some processes which they affect within the plant which is producing them. What we know is that these allelochemicals, if they are released into the environment, they can be toxic to other plants. So they are very toxic to the other plants, and we can exploit them to make herbicides. Or we can just use them in their organic form in weed control. We understand that some of these allelochemicals are toxic to insects, fungi, and bacteria. And they are actually used by the plant for defense. And so they are involved with the plant defense mechanisms. There's also scope to exploit these chemicals to synthesize insecticides or fungicides. These allelochemicals are also potentially poisonous to humans. So they could be toxic to humans. So we can have poisons out of these plants. Indeed, there are some plants which are really toxic, which can cause death. But at the same time, there are also plants with the medicinal properties. You could have had people now using zumbani. It's a plant with some medicinal properties. And some of these allelochemicals could be exploited to produce pharmaceutical products. Our interest is this route where we have plants producing allelochemicals which are toxic to plants, and these can be potentially exploited to produce herbicides. So this is our focus for this lecture. Now, what are allelochemicals? There are a number of chemicals which have been identified within the plants, and these are just examples. There are quite plenty of allelochemicals. Number one, we have got fumaric acid, 2 terpenoids, 3 syringic acid, 4 phenolic compounds, and 5 durin. Durin is actually produced by sorghum, as well as 6 sorghum is also produced by sorghum. Number 7 is caffeine, which is produced by coffee. If you have taken coffee today, you could have consumed the caffeine. Then we have got ricin, number 8. Ricin is produced by castor bean, and it's a very, very toxic substance. Atropine is produced by Datura stramonia, and atropine could be of medicinal value. We understand some traditional healers use atropine from Datura stramonia to treat asthma. Incidentally, atropine is also used to treat asthma it can be a pharmaceutical product, and is actually administered in hospitals. What you are seeing is a bottle brush tree, Aristenon citrinus. Bottle brush, probably because of its flowers. Now, if you know a herbicide which is known as mesotrione, it was actually, it started from here, from bottle brush tree. In 1977, the allelopathic weed control, which was happening near bottle brush tree, Aristenon citrinus, led to the identification of lepidopemon, a natural product, and this then led to the development of sarcotrione and mesotrione. It is mesotrione which is now being used to control weeds in maize. And the Zeneca agrochemicals developed this mesotrione. In Zimbabwe, it is being sold as galago, which can be used to kill weeds. You can either apply it pre or post-emergence in a maize crop. But what you should know is that mesotrione was developed because of the allelopathic effects of Callistenon citrinus. So that plant has this product. In Zimbabwe, we have Callistenon viminalis, which could be naturally occurring. The other Callistenon citrinus, I think it originates from Australia. Of course, we have people now planting it. I think it probably is an ornamental plant because of this flower. So plants produce chemicals via leaves or roots, which may interfere with the germination or growth of the other plants. So we do have live plants which can produce chemicals which have got an impact on germination and growth of the other plants. Now, when the plants die, they can decompose and produce allelochemicals, which can also cause interference with the germination, interference with the growth of other plants. And these chemicals, we call them allelochemicals. So here I've got a model of a plant. The plant can actually produce chemicals through volatilization. If you walk through a gum tree plantation, you may sense what is produced by the gum tree. Even other plants, you can actually, they can give a pungent smell. Like this weed, Tagetes minuta. Even the Datura stramonia. Now, after the rains, the allelochemicals may be reached from the leaves and they can fall to the ground, or the leaves may fall to the ground, then they decompose and release allelochemicals, which may be reached into the soil, or the roots themselves may be the source of allelochemicals. Now, in 1984, Rice defined allelopathy as the effects of one plant on another plant through the release of chemical compounds into the environment. So that was the definition given by Rice. This is a mango tree. It can also be allelopathic, especially the leaves. You can see under the mango tree, sometimes you don't get any growth, any plant growth. Of course, this could be due to shading, but the other effect is the toxic effects of the mango leaves. Lantana camara, which is a weed in Zimbabwe, which came as an ornamental plant, is also allelopathic. Here we've got a velvet bean. Velvet bean can also produce allelochemicals. Tithonia rotundifolia, with these flowers, can also produce allelochemicals. Now, there are two types of allelopathy. There's one which we call true allelopathy. This is the release into the environment of compounds that are toxic in the form they are produced. This is actually done by live plants. Then from dead plants, we have number 2, which is functional allelopathy, where the release of allelochemicals is a result of transformation by microorganisms. So the dead plants have got to be decomposed by microorganisms in order to release these toxic allelochemicals. So two types, one from live plants, true allelopathy, from dead plants, functional allelopathy. Now here we're going to examine the effects, what we notice on affected plants by these allelochemicals. You know, they are like herbicides. For example, you can see that a plant which has been affected by allelochemicals may have retarded germination rate, delayed or complete inhibition of germination. The plant may actually fail to germinate in the presence of allelochemicals. You may see damage on the root, which is necrosis, some cells turning brown, or you see brown patches on the root. The other symptom could be chlorosis. This is discoloration of leaves. We have noted that Lantana camara can cause chlorosis to some weeds like Biden's pilosa. Then there could be lack of root hairs as a result of coming into contact with the allelochemicals or leachates. Now, after absorbing the allelochemicals, this can actually cause a reduction in dry weights accumulation, or it can affect flowering, lower the reproductive capacity. They can cause wilting if they are in contact with the other plants. Or we can see reduce the plant population, presumably via reduction in germination. This is a paper which was done by Kwembeya at UZ, where he assessed the allelopathic effects of Lantana camara on Biden's pilosa and the permillet. Now, in this study, he used the petri dishes to study the germination of permillet and Biden's pilosa. So you would put in different concentrations of Lantana camara. This could be very dilute. Probably there's no allelochemicals here. As we move in this direction, these are dilute Lantana camara extracts. You are increasing the concentration. So at high concentration, the germination is actually reduced. Now, in these pots, he planted the permillet and incorporated Lantana camara biomass. On these pots here, there is high biomass of Lantana camara, and it tended to suppress Biden's pilosa as well as, but there's a bit of permillet image. But here, this is the control, where we have Biden's pilosa and permillet in these pots, and the concentration is increasing in this direction. Now, the Lantana camara residue effects on permillet, permillet was not really affected by the presence of Lantana camara, but Biden's pilosa, as we increase the biomass of Lantana camara, it tended to decrease, which means Biden's pilosa was sensitive to Lantana camara residues. This is the effect of different sorghum residues on Leucine indica, Acanthospermum hispidum, and Biden's pilosa. This was done in pots. This is actually a control. You can see in some of the pots, this is a control. You can see in some of the pots, some weeds were taken out by sorghum residues. Presumably, this is the effect of durin from sorghum, because this was sorghum biomass which was incorporated. Here we've got a paper by Runzika, who was studying, green manure covering crops. We know they can supply nitrogen, but at the same time, these green manure covering crops are endowed in allelochemicals, which can also kill some weeds or other plants. So it may be possible that we may find a green manure covering crop supplying nitrogen and at the same time controlling weeds. Another study was done by Dr. Rugare on the effects of green manure cover crops on the germination of Biden's pilosa and the Leucine indica. If you check on the internet, you may get his thesis, his doctoral thesis on allelopathic effects on the green manure cover crops. Of course, it started from here. Then we had a bigger study here. Now, there's also a paper by Murimwa, which examined the sorghum allelopathic effects on sesame. This is an experiment showing the allelopathic effects of the green manure covering crops on Erosine indica, Acanthospermum hispidum, and the Biden's pilosa. Now let us look at the processes which are involved in releasing allelochemicals by donor plants. Donor plants are the ones which produce allelochemicals. Now, after rainfall, we can have what are known as the runoff or leachates from the leaves and the stems. So the rain can actually remove allelochemicals from plants which fall to the ground. This was noted on a tree known as black walnut jugulans nigra, which was inhibiting vegetation underneath it or near it. Actually, this is what caused the people to investigate allelopathy. Or the allelochemicals can volatilize from plants into the atmosphere. Sometimes we can detect the smell of some plants. This could be probably due to the volatilized allelochemicals. For example, this can be noted with eucalyptus species, which we are familiar with in Zimbabwe. Another plant is Artemisia californica in other places. Can also volatilize allelochemicals. This is an example of a plant, a tree which can produce allelochemicals. This is the gum tree, which can also produce allelochemicals. Now, the release of these compounds, which are phytotoxic, could be a result of decomposition, for example, of wheat mulch, which then suppress yields. Another source could be the roots. The roots can actually release these allelochemicals and suppress weeds. In fact, we have got a rice plant which was bred, which releases allelochemicals which can kill plants. So in that rice variety, you don't actually need to weed it. I think it was developed at Erie, where they are doing some rice research in Asia. It would be interesting one day if we could get a plant in Zimbabwe which produces these allelochemicals and kill weeds. Now I want to look at the factors which can affect allelopathy. Now, crop varieties. Allelopathy is controlled by genes. So you may find that different crop varieties may differ in terms of how they produce these allelochemicals. There is also specificity, or you may call it selectivity. You may find that some allelochemicals may kill some weeds may actually survive. So they don't equally affect all of the plants. Some survive, some will not survive. So there is specificity. We also have crop-on-crop effects, where we have residues from allelopathic crops inhibiting the germination and the growth of the following crops, as well as weeds. The environment, especially soil fertility, pests, and the diseases. Now, low fertility may increase the production of allelochemicals, because some allelochemicals help in the acquisition of nutrients. How pests and diseases also affect is that once a plant is attacked by pests or diseases, it may respond by producing allelochemicals, probably as a means of defense mechanism. Then radiation, water stress, and temperature, these may also induce production of allelochemicals within the plant. Then age of plant organs, the age of the plant may also determine its capacity to produce these allelochemicals. So allelochemicals production can be affected by a number of things. Here we just want to go through the mode of action of allelochemicals. By the way, there are many allelochemicals. So they actually act like herbicides. So this list which I have about the mode of action is what is thought these allelochemicals may be doing in the plants. But this is just a general picture. Number one, allelochemicals can cause cell division and cell elongation. Probably they have some sort of hormonal effect. They can cause phytochrome-induced growth. They can affect membrane permeability. They can affect availability of nutrients, soil, phosphorus, and potash. They can also act on gas exchange, affect processes of photosynthesis, they could affect respiration and also mineral uptake. 7 and 4 could be associated. Here you can see I'm not giving actually examples, because I think it is in this area where there's a lot to do. A lot needs to be done actually in terms of research. Protein synthesis is also thought to be affected. Change in lipid and organic acid metabolism. Because some plants turn white, chlorophyll is affected. So number 10, it is thought that there could be inhibition of the porphyrin synthesis. Porphyrin is associated with the chlorophyll. Because sometimes you see plants wilting, probably these allelochemicals affect water relationships. For example, they could be blocking the xylem vessels. And this can lead to shortage of water within the plant. They are also thought to stimulate or inhibit certain enzymes. So some of these are largely a hypothesis, which is trying to explain what the allelochemicals are likely to do within the plants. But some of these they have to be established. I'm going to go through some studies which have been done to demonstrate allelopathy. I shall start with some studies which were done in Zimbabwe to demonstrate allelopathy. We've got sorghum and pearl millet, which were found to have a negative effect on Bidens pilosa, Acanthospermum hispidum, and Leucine indica. These were actually suppressing these weeds. Green manure covering crops, for example, Mucuna, Clotularia, were found to suppress Bidens pilosa, Acanthospermum hispidum, and Leucine indica. Then we had Lantana camara, which was found to suppress Bidens pilosa, but he could not suppress pearl millet, which means we could use Lantana camara to control weeds in pearl millet. Now, Cyperus rotundus, which is a serious weed, it may compete with various crops. It was found in cotton to suppress cotton. In fact, Chivinge, who noted it, found that at Henderson, some cotton was standard in areas where there was a Cyperus rotundus. He also noticed it, I think, in Mid-Rusabi, where cotton was not performing very well in some fields. And he did a study which established that indeed Cyperus rotundus had some allelopathic effect on cotton. It was also established that at Grasslands Horticultural Research Center that Cyperus rotundus was suppressing the growth of horticultural crops. Tomato, squash, onion, cabbage. This was done by Kawisi. Kawisi was working at the Horticultural Research Center, and he noted this effect in the fields. She subsequently made a study which established that indeed it was Cyperus rotundus which was suppressing these crops through allelopathy. Then wheat straw was also found to be allelopathic to some weeds. At Henderson Research Station. Then sorghum exudates were found to stimulate striga asiatica. We'll talk about it. But multi-purpose trees were actually suppressing sorghum. There was a field study which was done in Chimanimani district by a student. It was interesting to note that some crops were associated with a very low number of weeds. For example, pearl millet. As well as the cowpeas. It was thought that probably these crops were suppressing weeds via allelopathy, and indeed, in some studies, pearl millet has been established to suppress weeds via allelopathy. Now we do have a coffee which produces caffeine. Caffeine was noted to suppress Amaranthus spinosas. Wheat mulch, which produces ferulic acid, was also found to suppress Hypromia species, but sometimes it had a neutral effect. Some soybean extract suppressed the Inderata cylindrica. In some places, sunflower was suppressing Phalaris minor. There are actually some farmers who think that if you grow sunflower in a field, it tends to suppress weeds. But I think we need to establish it in Zimbabwe. But farmers, always say that if you grow sunflower, you suppress weeds. Now, pigeon pea, velvet bean, doricos, again sunflower, soybean, lablab, and radish were found to suppress this downy broom. It's a weed in the US. And then we had cowpea and the sunflower, which also suppressed Erosine indica and the Amaranthus hybridus. So sunflower, here it was noted here and also there. probably sunflower could be suppressing, could suppress weeds. Well, there are also weeds which can do the same thing, which is not really good. Ageratum cordyceps could affect wheat. Wild oats could also affect wheat via allelopathy. Cynodondactylon could also affect crops. Carrot, cucumber, maize, onion, radish, tomato, and sunflower. I remember I showed you a slide of a field with very short maize growing in association with the cynodondactylon. And we thought probably the sudden, this sudden sort of reduction in maize heights compared to other plots could have been caused by allelopathy, because there was a dramatic, impact of cynodon on the maize. But we may need to investigate this. Then that Datura stramonia, which is a weed, was also found to affect sunflower via allelopathy, and the Datura stramonia produces two products, scopolamine and diazomine, as the allelochemicals. Then crop effects on crops. We have got sorghum and rice. If you grow sorghum, rice, and rotate it with maize, maize could be affected, by the allelochemicals produced by sorghum and rice. And these are the list of some of the allelochemicals: benzoic acid, p-benzoic acid, vanillic acid, garlic acid, which can affect maize. Mung bean, if you rotate it with soybean and lettuce, it can also have an impact on these two crops through allelopathy. So we now know that some crops or some weeds can produce allelochemicals. Can we use allelopathy as a weed control strategy? Yes, there are opportunities to do this. First, in crop rotations, cover crops, mulching, intercropping, or we could actually extract some allelochemicals and apply them to weeds. Well, these are some of the references which you may read about allelochemicals, the application of allelopathy in weed management. I think these two references are good. So let us look at how we can deploy allelopathy in crop rotation. First, we need to identify crops which can potentially produce these allelochemicals. We've got sorghum, peru millet, wheat, cowpea, and sunflower as potential crops. So we could grow them, and we should expect a reduction in in the wheat problems. Probably we should do this under conservation agriculture, where there's a potential to have a lot of weeds. But we need to screen these crops so that we get the best candidates to deploy in crop rotation. So this is permanent, which can also reduce weeds. Sorghum can also reduce weeds. You know, there's a lot of sorghum which is being grown by smallholder farmers, especially in the low veld. In fact, there's an area where there's a proposal to grow the same, and the farmers, they are preferring to grow sorghum, which they are already selling to the I think they are selling to those who are brewing beer, to a company which does brewing of beer, and they are making a lot of money from sorghum. Probably in those areas, weeds are being suppressed by the sorghums. Wheat can also produce allelochemicals, telluric acid, which can suppress weeds. Cowpeas, they are also reported to suppress weeds via production of allelochemicals. So these crops, we could fit them in crop rotations. Sunflower is also a potential crop to use to suppress weeds. Then cover crops, like these green manure cover crops, like sun hemp, Crotalaria janisi, cowpea, velvet bean, and lablab, paparia. You know, we can just grow these crops or these plants in some areas just to improve soil fertility. And at the same time, we reduce weeds, because they can also increase nitrogen levels in the soil. But growing cover crops may cause farmers to forego certain fields in a cropping season. And probably farmers may not like this. But in the case of cowpeas, maybe cowpea may be attractive because they can use cowpea during the same season. They can get grain from it, improve soil fertility, and kill weeds at the same time. Maybe it could be attractive. This could be cowpeas, which can suppress weeds. Mulching. Now, mulch is the spread of crop residues on the soil surface to suppress weeds. Wheat mulch can do the same effect. In fact, for wheat farmers, you may notice that after wheat in October, we get some rains in November, And usually the wheat fields may not have a lot of wheat due to the allelopathic effects of wheat. This is maize mulch in Gokwe. Maybe maize mulch is not very effective in suppressing weeds. I think this is a legume crop. The farmer is trying to explain what is happening here. Soon after harvesting maize, he had to grow this bean with late rains. So again, this is maize mulch. But in some cases where you have livestock production, this mulch, instead of mulching, this could be fed to livestock. Intercropping. Now there are opportunities to intercrop maize with sunflower, maize with cowpeas, maize with the groundnuts, maize with bambara nuts, or sunflower groundnuts, sorghum groundnuts, sorghum bambara nuts. Now this system may have crops like sunflower, cowpeas, sorghum, which can produce allelochemicals, which will kill the weeds. However, there's a need to screen these crops to evaluate their effectiveness. But there are opportunities to deploy allelopathic crops via intercropping so that we can increase crop production and minimize weed damage. Here we've got an example of intercropping. You can see where these farmers are, young farmers, in fact, with the maize and cowpeas. And cowpea can be allelopathic. It can suppress maize, and it is planted between maize rows. And here it may not pose serious competition to the maize. Now, allelopathic water extracts. We can actually produce extracts from known plants, from some plants which are known to be allelopathic. There could be opportunities to mix these allelopathic extracts with some low doses of herbicides. Some have actually done this. I think there's a paper by Dr. Rugare on this subject. Now, we have been discussing the applications of allelopathy on weed control. Here we should now look at the methods which are used to study allelopathy. Now, allelopathy can be examined in 3 stages. You can start with the laboratory study, then you do a glasshouse study, then a field study. And you can test these allelochemicals in these 3 stages. Now, in a laboratory, what you may because you want to demonstrate allelopathy, or you want to check whether some plants produce allelochemicals. You need to harvest leaves, stems, roots from the plant which is potentially allelopathic. You grind these parts, maybe sieve. You can, for example, get 10 grams of the plant parts, put it in distilled water, allow it to stand for 24 hours. Maybe you use 1 liter of water for this. Then by the end of 24 hours, you'll have extracted the allelochemicals. This could be probably, for argument's sake, 10% solution. And you can apply, you can do now germination tests using this solution. In this case, you measure germination percentage. You also measure the length of shoot and the root, or radicle and the plumule. This slide is showing plant extracts from green manure covering crops, which were tested for germination. These are extracts from Lantana Camara. So these are petri dishes with Biden's pydosa. And you can actually, probably someone was applying some allelochemicals or leaf extracts to these petri dishes. You can see a lot of petri dishes where somebody is working on plant extracts. This is actually Alan Kwembe. You can't see who wrote a paper on allelopathy. Then there's a need to do glasshouse experiment where test plants can be planted in pots, and allelochemicals are applied on those plants. Here you can actually incorporate biomass in the soil, or you simply spray the plants and then monitor growth and later on harvest biomass. Like what was happening here, biomass of sorghum residues, which were being evaluated for allelopathy, were incorporated in these pots. Then these 3 weeds were planted. And of course, things like emergence was monitored. Now, you can now move on to field experiments. Let's suppose you have noted from your glasshouse work and the laboratory, you have identified some potential plants which are allelopathic. You can test them in the field. You can grow these allelopathic plants under field conditions. There are 2 ways in which you can do this. It's either you can incorporate the biomass in the fields, or you can spray the allelochemicals. You need to have some rates of allelochemicals to be tested. And of course, this should be, a typical experiment with good replications, randomized treatments, and so forth. And the type of data which you may collect could be emergence of the crop, weed counts, weed biomass, crop phytotoxicity, and grain yield. Now, if you find that there are some allelopathic plants which can potentially be used to control weeds, you may be interested in the chemistry. You want to find out what is actually causing this effect. So you need to extract the allelochemicals, identify them. Now, there are various methods which can be used. There are simple methods which use paper and thin layer chromatography. You can also have facility for the HPLC. And these days, they are now using the one which is popularly being used is the LC-MS/MS to identify the allelochemicals. This is the HPLC, which is high performance liquid chromatography, which can actually identify the allelochemicals. These are just simple chromatographic methods where you have these three dots. This is where you are testing your allelochemicals. You have got a solvent which moves these liquids. You will have a standard. So after a while, these dots migrate upwards. And it can tell you something about what is contained in the extracts containing allelochemicals. This is sogorion, one of the allelochemical produced by sorghum. This is caffeine, an allelochemical produced by coffee. And these are flavonoids. These are commonly produced by most plants. So we have somebody who is now working on the allelochemicals. In fact, there were a number of sorghum plants, and what he is simply doing is extracting the allelochemicals from young seedlings of sorghum plants. There were several sorghum plants from, I think, from SADC region, which were being examined. Again, here's a bath where the extracts are actually concentrated. And there are some of the concentrations. And this was actually used after, this one is The one in the lab is Dr. Tibugari, who was extracting allelochemicals from sorghum. He had to use the LCMS for determining sorghum. So it's Dr. Tibugari doing the extracts. Here concentrating the extracts before sending them to the LCMS. And these are some of the results, showing peaks of sorghum. Now, these are the varieties, sorghum genotypes from Botswana. Remember, he was doing for the whole SADC. Now, these are some of the, sorghum concentrations. The idea was to try and find or identify a sorghum which can produce a lot of these so that it could be recommended for wheat control. So you can see these are the variable, You can see this sorghum here produce a very little amount of sorghum, but this one quite a lot. So this is some of the data you was getting. He also looked at the Zimbabwe wild and the sweet sorghums in search of this sorghum. And there was this wild sorghum with a bit of, high sorghum, but in contrast to the other wild sorghum with very little production, it appears to be genetically controlled. If you are interested in that story of sorghum, you could get hold of Dr. Tibugari's paper on the quantification of sorghum in sorghum accessions from 8 Southern African countries. But this was actually done here at the University of Zimbabwe in the weeds laboratory. You can also have a look at a paper on green manure cover crops by Dr. Rugari. In fact, Dr. Rugari also used this he also used this same technique to check these allelochemicals in green manure covering crops. Here we've got jack bean and velvet bean. Probably jack bean had what is known as gene stain. Otherwise, it didn't have a lot of these other allelochemicals. But jack bean had camphorol. It had naringenin. It also had a lot of rutin and also jade stain. This atropine was not detected in all of these three, these two green manure covering crops. Atropine is mainly found in Datura stramonia. So these are some of the, compounds which were detected in Dr. Rugari's study, searching for these allelochemicals. So these are some of the compounds. Now, there are references which you may have a look at. Like this one, Putman, he wrote a lot of things around allelochemicals. He is actually talking about the potential of allelochemicals to be used as herbicides. And also Juk is talking about the methods of demonstrating allelopathy in crop weed interactions. Also, this other paper, you can always follow these papers. Some are useful. Some papers, you may not like them. So let me summarize what we have just talked about so that you see the path of this discussion. We discussed the allelopathy. We said it has got two. We've got there are two parts: true allelopathy and the functional allelopathy. True allelopathy, we have got a live plant which is producing allelochemicals. And functional, the plant has to die and decompose in order to produce allelochemicals. Then we have got examples of allelochemicals like sogorion, which is produced from sorghum. And there was a survey of sogorion in the SADC, in these sorghums by Dr. Tibugari. This durin is produced from dead sorghums during probably, I could be right, during decomposition. You may have to check. But this one is from a live sorghum. Where are these allelochemicals coming from? They are coming from leaves, stem, and roots. We also discussed the factors which affect, allelopathy. How the plant produces these allelochemicals could be probably in response to its defense. Maybe the plant has been attacked by an insect, or it could be a disease, or it has experienced a drought, or it could be through nutrition, and it produces these allelochemicals. So they have, in a way, they have a role in the plant survival. Then effects of allelochemicals and their mode of action. This is what we see when a plant has been affected by allelochemicals. It could be low germination or discoloration. And what causes that could be, maybe the chlorophyll has been modified and so on. So we discussed how allelochemicals are likely to produce these symptoms. But more research needs to be done, because most of it are just assumptions. We are trying to learn from the real herbicides and try to apply to these allelochemicals. We also went through a sort of a review of how allelopathy was demonstrated in Zimbabwe and elsewhere, just to give you a picture of what allelopathy can do. Then we examined the application of allelopathy in crop rotations, cover crops, intercropping, mulches, and also the use of extracts of allelochemicals. Then we looked at the demonstration of allelopathy in the laboratory, glasshouse, field tests, and the actual identification of allelochemicals, because it will be necessary to identify these allelochemicals. Maybe they may end up being used as herbicides. Herbicides could be synthesized from these products. So this is it on the allelopathy. We have to stop here. Are you still there?
Yes, Doctor.
At almost dead, so tired.
Yeah, I was almost lost. So this is it about allelopathy. What you only need to know is that plants produce allelochemicals, which can affect other plants. In fact, they can also produce toxins. Because they produce allelochemicals, there's a potential to produce herbicides. And in the beginning, I showed you a plant which is now being exploited to process herbicides, which are currently being used. And of course, there are other examples where allelochemicals have been demonstrated in Zimbabwe. Anyway, there's this Dr. Tilugari who did a lot of work, with the sorghums, trying to identify the sorghums which can produce a lot of this sorghum. The intention is that if we come up with such a sorghum genotype, we can recommend it to farmers so that they actually control it using that sorghum. Any questions? On the allelopathy?
From me, I don't have any questions. But ndokumbirawo kupuwa mamwe maPowerPoint asiri marecordings emalectures so that I can go through. I'm facing challenges yekuma-download but ndinaione offline. Hey, data racho rinenge riri slow.
Okay, yeah, what I do, when I send you recordings, I also send you original PowerPoint.
Handimaoni sei.
Uri kudownloada uri phone?
No, ndikuda hotspot laptop ndo-downloada saka ndaona mavideos so far.
On my video. Okay, I will try to send it, then we communicate.
Okay.
to see what is actually happening.
Yes, the PowerPoint is in a recording.
Yeah, I'll do that. In fact, after this, I'm going to send you the PowerPoints on the competition and allelopathy.
It's okay.
Okay. Thank you.
Thank
But when you try to download, you can also send me a message.
Okay.
It's okay.
Indicate whether you have actually managed to download.
I will tell you.
So tomorrow, yeah, we will have a lecture on the parasitic weeds. I'm intending to finish tomorrow this part. We may leave Friday free, I am not sure.
Okay, Doc.
Yeah. But I will communicate with Dr. Rugare after tomorrow's lecture.
Okay.
Then again, tomorrow, later on, I will post some assignments.
Okay.
Some of these assignments may be a sort of a question model so that you know what sort of what to expect when we set exams. Then I
Okay.
may give you sort of a practical where you are. Can you access a field?
I can.
I can.
What research board there? I think there are plenty of fields there.
Yes, I can.
But if you are writing in the city center, then there can be problems. We will see what we can do.
Okay.
Yeah.
I don't know, Isaac, we can always make a plan to itateze kunoko.
I think that would be wonderful.
It's okay.
If you could do it at Kutsaga Research Station, right?
Only so.
Yeah, you will do something which is, which will not cause problems there.
Okay.
Yeah. So we will stop here. I will process the video and the notes are already there. So I will just post them.
All right. Thank you, Doc.
Okay, thanks very much.
Good day.
All right. Good day.
All right.
Okay. Sharp.